A question that keeps coming up is how we prepare parasite samples for DNA analysis. Several of us led a workshop in Argentina a couple months ago where we trained wildlife vets and infectious disease experts how to do exactly this. By the way, the workshop happened at Iguazu National Park, which is one of the wonders of the world. 10 out of 10. Highly recommend it. More and more people are trying to use DNA to identify parasites these days, but they're stunned to learn how few of these parasites we can actually identify this way yet. At the workshop, we found out that there's still a lot of parasitologists out there who aren't even sure if they're collecting specimens in the right way for this kind of work. Here, we're going to share the critical first steps of how we prepare these specimens for genetic analysis. It's all part of our series of videos on how to succeed in molecular ecology. We've already shared the origin story of some special samples from parasites that we have in the lab based on an epic trip that a wildlife veterinarian from our group took into the Chaco of Argentina, ran into some wildlife poachers along the way. And we shared the much anticipated unboxing moment when the samples finally, after months, arrived in our lab. Now, there are many different ways that people try to preserve parasite specimens for analysis, and the traditional way is to sink them in formalin. Formalin is good if all you want to do is look at the parasites under a microscope, but it's really bad for any genetic analysis. Formalin destroys DNA. Another option is to use ethanol, which is what we do. Ethanol is really good at preserving DNA, and it's also very good at preserving the morphology of specimens if what you want to do is connect the DNA to taxonomic assessments, morphological assessments of the specimen under a microscope. And the way ethanol works to preserve DNA is it basically forces any water, any moisture out of the tissue of the specimen and allows the DNA to be preserved that way. Water is the enemy of DNA. By forcing the water out of the tissue, the specimens have DNA that can last for a long time. Now, the first thing we have to do is rinse the specimen, and rinsing the specimen in this way allows us to remove all the ethanol that's been preserving the specimen so that we can proceed into our DNA extraction. We need to remove the ethanol, we need to rehydrate the tissue, and so that's what they're doing here.
Yeah, you finally got a big one.
We find that rinsing the specimen with water in this way is so much faster and easier and safer than leaving the specimens out on a bench to evaporate, to dry for long periods of time. It's also not a fire hazard. Don't let ethanol accumulate in your HEPA filters. It can cause an explosion, and you don't want that. We find this kind of quick rinse with Millicue water is all it takes to remove enough of the ethanol and give us good, fast, effective results. Now that the specimen's all clean, we can prep it for extraction. And a word of warning here, the editing is going to become a little bit funky because people were talking about, like, boyfriends.
It's all getting recorded.
And I don't want to embarrass anybody, so we'll see how it goes.
We'll edit it out.
So the parasite they're working with here is an acanthocephalin. It's sometimes called a thorny-headed worm, and it is a specimen that our students actually found at the workshop in Argentina inside a big-eared opossum, a South American big-eared opossum. Parasite experts are going to notice that it has these thick cuticles, and those we find can make it very challenging to get a good DNA extraction from specimens like this. So we've worked really hard to come up with strategies that work efficiently and consistently. So what they're talking about here is basically how to divide up the specimen.
Should I cut him into a bunch of pieces and then or should I just cut off a little bit of him for now?
Probably don't need all of them.
Yeah. Like a quarter of him, maybe?
Maybe even half. He's pretty big.
He's chunky. Maybe
See what a quarter looks like then.
Okay.
This is a big, chunky specimen, and we don't need the whole thing. We often recommend like a pea-sized amount of material going into the DNA extraction. That's just for the kits we use. Different kits have different expectations for the amount of input, and so you should always check.
He's hollow, ew.
I mean, he's a tube.
Yeah, that's pretty disgusting. Awesome.
We've tried a lot of different DNA extraction methods, and the one that we find works best for these kinds of parasites is a simple enzymatic digestion with proteinase K, common type of strategy that you'd find in like a Qiagen blood and tissue kit. We've tried alternatives like mechanical lysis, bead beating the specimens, And we just don't find that it's very effective at breaking through these tough cuticles. If you're someone who likes working with liquid nitrogen and mortar and pestle, you do you, that's fine. For us, we find this proteinase K digestion works the best.
Awesome.
I would cut them up a little bit more though.
Oh, cut them up.
Yeah.
We still want to increase the surface area of the specimen so the enzymes can do their job, especially with big specimens like this.
It looks kind of
I'll put the rest
chewy, for
back.
a lack of
Does
a
that look
better
like
word.
enough for?
Yeah, I think so. And then that piece that you cut off, see if you can get it into like
A little bit.
4 pieces maybe.
Yeah.
I feel like the enzyme's been doing a pretty good job.
And then we'll incubate it overnight, which is a good bit longer than many of the kits will tell you need to. But we have found through trial and error that longer digestions work better for these kinds of specimens.
Carnage.
How's that look?
Pretty
Messed up
gross. Yeah.
I don't know, should I be blurring this part out? I think it's gross too, but these specimens are so important
Love it.
if we're going to find the genomic opportunities that help us fight parasites more
You said
effectively.
we have to name him.
That was Mr. Big.
Oh, yeah, he was.
For this kind of work, we try to be very clean in the lab so that little blips like this don't matter very much. We have other videos that show you more about our standard sort of clean up and clean down protocols that we find help maximize the chances of success while minimizing the risk of contamination in the lab.